Hello, in this lecture, we'll talk about tryptophan operon, and this class is a continuation with the previous class, which deals with prokaryotic gene expression and overall gene expression regulation. So, this class is all about operon or tryptophan operon. Now, uh, operon, unlike operon, is an um, inducible. It's not an inducible operon, it's a repressible operon. So operon was an inducible operon. That means operon by default it was off, and on demand, whenever lactose is present, operon was switched on. So here we before we go and learn what are the uh, basic aspects of the tryptophan operon and all, before even that, we should talk about operon and operon comparison. So operon versus tryptophan operon. So operon is an inducible one, whereas tryptophan operon is a repressible operon. Now, what we can understand, tryptophan operon is simply, so we just tryptophan operon helps in biosynthesis of tryptophan. Now, question is, why bacteria need tryptophan? Bacteria are cells. They need to meet their metabolic demand, they need to reproduce, and they need to produce a hell lot of enzyme. In order to produce a hell lot of enzyme, they need to produce amino acid. So for that, one of that amino acid is tryptophan, and tryptophan is an important component of many proteins. So tryptophan need to be synthesized all the time because they need tryptophan for building proteins. So we just look at the tryptophan operon gene configuration. So here is the operon gene. So again, what is operon? Operon is a set of genes controlled by a single promoter. Here, a set of genes TRPA to TRPE genes are controlled by a single promoter or TRP promoter. So that is why this is known as operon. So what is the normal function of this operon? These operon give rise to 5 different enzymes. So they give rise to specific enzymes, and all these 5 different enzymes are actually part of a biosynthetic cascade which converts corysmic acid. Now, corysmic acid is the raw material from which tryptophan is produced. So, and this process of conversion of corysmic acid to tryptophan is stepwise, and in several steps, these enzymes play a role. So, simply, tryptophan biosynthesis requires these operon gene products. So, if operon gene products are there, tryptophan biosynthesis would be aided. If tryptophan biosynthesis uh, regarding tryptophan operon genes are not transcribed, then tryptophan biosynthesis won't happen. So we talk about when this operon is repressed and when this operon is on. So this operon is repressed when there is plenty of tryptophan in the bacteria itself. So it's kind of an on-demand situation. When you have too much tryptophan, the cell don't want to waste its energy by producing more tryptophan, right? So then what it would do is it should switch off the operon and try to save its energy. But whenever the tryptophan level is going down, then the operon would get switched on, right? In order to produce more tryptophan, and such that that tryptophan could be used to produce many other amino acids. So that is pretty simple. Now we look at a situation of high tryptophan versus low tryptophan and see that how high tryptophan versus low tryptophan affects the operon. Now here is the thing. So whenever the tryptophan level is low, so we assume a situation that tryp level is low. Whenever tryptophan level is low, the operon gets switched on, right? What happens? Like lac operon, here also you have a trip repressor. Let's name it trip R, that give rise to specific mRNA and which is transcribed into tryptophan repressor. Now tryptophan repressor is an apoenzyme normally, but without the tryptophan it cannot bind to the specific operator region of the trip operon and cannot suppress the tryptophan biosynthesis. Only when tryptophan is present in ample amount, it would produce a holoenzyme that can in turn bind to the promoter and block the transcription. So whenever tryptophan is low, there is not abundant tryptophan to bind to this apoenzyme or to activate this repressor. So in absence of this repressor, tryptophan genes, trip operon genes are produced, and as a result, all the protein products are also produced, and the end point of that is production of tryptophan from chorismic acid. Now we'll look at a situation where the tryptophan level is high. So we'll look at a situation when the tryptophan level is high. So whenever tryptophan level is high, what happens? The operon would be switched off, and we'll look at the mechanism by which the operon is switched off. So whenever tryptophan level is high, the operon would be switched off, and we'll look at how the operon would be switched off. So whenever the operon is switched off, transcription from all these genes are turned off. So none of the protein products are produced, and as a result, none of the enzymes that convert chorismic acid to tryptophan doesn't happen. So in absence of too much tryptophan, what happens? Our trip repressor is always produced, but trip repressor is normally a aporepressor. Without tryptophan binding, it cannot get activated. So when tryptophan binds, it gets activated. This repressor gets activated, and as a result, this repressor binds to the specific operator region in the trip operon, and as a result, what happens? The RNA polymerase cannot simply bind to that promoter region, and as a result, RNA polymerase cannot bind and the transcription don't happen. So whenever tryptophan level is up, the operon is switched off. So these two situations tell us that bacteria is producing tryptophan on demand. So whenever there is a demand of tryptophan producing, bacteria would switch on the operon and produce the tryptophan. Now whenever there is plenty of tryptophan around, it don't need to produce and waste energy by producing more tryptophan. Then it can switch off the operon for a while, and whenever the tryptophan level is depleted, it can switch on the operon again. So that is how cleverly bacteria can optimize its energy consumption and also regulate the expression of the tryptophan metabolism gene. Now, this tryptophan operon is controlled at several levels. So whatever example I've given you, it's a repression of the transcription that I've talked about in presence of tryptophan. And the repression happens by the trip repressor, which is becoming a hollow enzyme in presence of the tryptophan. But it's an apoenzyme and inactivated state in absence of tryptophan. Apart from that, there is another mechanism by which the trip operon genes are regulated. This is known as the attenuation, transcriptional attenuation. So let's just take a moment to look at what is transcriptional attenuation. So here is our tryptophan operon genes. So we look into tryptophan operon genes. So we have trip A, trip B, trip C, trip D, and trip E. And here we have the tryptophan promoter. In the promoter region, what happens is our RNA polymerase is binding. So in the promoter region and the operator region, there is a region which is known as trip L that codes for a leader RNA. So let's say the RNA polymerase has bound on the promoter, and now it would move forward. It want to move forward and transcribe the gene in this direction, right? Now what happens is the newly formed mRNA is producing. It is known as a leader mRNA, the initial part of that, known as the leader mRNA. Now it would move forward. Now this leader mRNA has four particular regions in it. So we zoom into the leader mRNA. So here is our leader mRNA. Now leader mRNA has few zones. This is zone 1, zone 2, zone 3, and zone 4. These zones are complementary to each other, such that what can happen is zone 1 and zone 2 can pair with each other, forming a hairpin. Zone 3 and zone 4 can also pair with each other and forming another hairpin. So hairpin formation can happen. Also, regions 2 and 3 can pair with each other to form a hairpin. Now, this configuration on the right hand side is known as anti-attenuator, whereas the left hand side is known as attenuator loop. Now this attenuator loop is the structure for which the trip operon gene are not transcribed. So you can expect that whenever tryptophan level is high, then attenuator loop is formed and the transcription of the genes of the tryptophan operon does not happen. But let's just take a moment to think that why this attenuator loop is first of all formed, and second, if the attenuator loop is formed, why does the transcription does not happen further? Why does the downstream genes are not transcribed? So the answer is here. So the RNA polymerase has to traverse from the 5' to 3' direction in order to transcribe the downstream genes. Now, whenever the attenuation stem loop is formed, attenuation stem loop formation, it has been correlated with high levels of tryptophan. So whenever there is a high level of tryptophan, attenuator stem loop is formed. And why does that happens? I'll show you in a moment. So let's assume the tryptophan level is high, and then you have two type of stem loop formed. So region 1 and 2, region 3 and 4, they paired, and in the attenuator configuration, there is a small region which is rich in uracil. This region, what you can see is actually, this region is basically rich in uracil. 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 This That means it's not strongly attached with the transcription bubble. So as a result, RNA polymerase, before it can move forward, it might fall off from this region. It might fall off from that region itself. So now, how fast? So we have to understand that the transcription is not an isolated event. Transcription is always coupled with translation in case of a bacteria, because the bacteria don't have a compartment, when specific compartment where the transcription is happening. So here what we can see is the RNA polymerase is proceeding, and from the other hand side there is a ribosome which is sitting on it and producing the specific proteins from the RNA, leader RNA. Now when tryptophan is high, when tryptophan is at a high level, what happens is there is a sequence between 1 and 2 region, which has multiple tryptophan codon. So when tryptophan level is high, amino acyl tRNA for the tryptophan is there. It can easily bind there, and the and the ribosome speed on these particular RNA is pretty fast. So ribosome would nicely translate all those regions. Now, as a result of that, what would happen is this attenuator stem loop would be formed. But let's just look at a situation when the level of tryptophan is low. Now, when the tryptophan level is low, 1 degree of regulation is formation of the repression of lack of repression of the operon via the repressor, and in less tryptophan condition the repressor is inactive, so there is nobody to repress the operon. But apart from that, what happens is the ribosome, which is on the newly formed leader mRNA, won't move with a great speed, because imagine the amino acylated tRNAs which codes for tryptophan in this specific sequence which is required are not abundant right now, because itself the tryptophan level is low. So the amino acid, though the tRNA which would be charged by tryptophan is also low, so that cannot come quickly. As a result, what happens? The ribosome slows down. When the ribosome slows down, it is lingering around the region 1 only. Region 2 and 3 are free, and as a result, what happens? As a result, what happens is anti-attenuator loop is formed, because the ribosome is lingering around region 1. So ribosome would be somewhere here. So the region 2 and 3 are free, so they can complementary base pair with each other to form this anti-attenuator stem loop. When this thing happens, the ribosome can, the RNA polymerase don't slip off, and it proceed towards the 5' to 3' direction, and as a result, what happens? This mRNA of the tryptophan operon is produced, which would ultimately give rise to specific enzymes which are required for tryptophan biosynthesis. So this makes sense, because whenever the tryptophan level is low, there are two combination of mechanism. One mechanism is definitely that negative repression. That means repression is released, so the repressor is inactive in less amount of tryptophan, and as a result, the tryp genes are transcribed. Not only that, we also seen that the attenuation of the transcription does not happen when the tryptophan is low, whereas the transcriptional attenuation happens whenever the tryptophan level is high. Then the transcription attenuation happens. Then in case of transcription attenuation, the downstream genes are not transcribed, because after a point of time the RNA polymerase slips off from the DNA and it cannot transcribe the downstream genes. That is how by the combination of transcriptional attenuation and repression, negative repression, this whole repressible operon is regulated, and thereby on demand bacteria can produce its tryptophan uh, tryptophan amino acid, which it can use for other purposes like protein synthesis or enzyme synthesis. So that's how we can understand how a bacteria can strategically maximize its energy and not waste its energy by producing something which is already there, already can be used. So in that regard, E. coli is also very intelligent. So I hope you like this video. If you like this video, give it a quick thumbs up. Don't forget to like, share, and subscribe. Thank you.